Stanford University.
Please welcome Dr. Irv Weissman.
Thanks, Bev. So this field got its start with a tragedy, the bombing of civilian populations in Hiroshima and Nagasaki. The way it happened was that people who were not killed by the force of the bomb or the fire after the bomb, and who didn't die in the first 5 days, died at about 15 days after they were radiated. And it was this radiation syndrome that led people to try to figure out what went wrong. Well, mainly what went wrong is with this blood-forming system. The dose of a radiation that's the lowest dose that kills by wiping out the blood-forming system. Many, many scientists were then funded after World War II to try to figure out what the radiation syndrome was. One of the neat experiments they found when they irradiated mice, now not people, was if they put a lead shield in front of any bone, the animals didn't die of the radiation syndrome. Later it was figured out that it was the cells within the bone marrow, that were the essential cells, and people started transplanting bone marrow from one mouse that was unirradiated into a mouse that was irradiated, and it would save them. For the longest time, however, a few key figures in the field who were important people and who said, well, the bone marrow that you put in releases radiation repair factors and saves the bone marrow that was there. That was exactly wrong, but it was the conventional wisdom. Then 3 groups of people said, well, what if I put in bone marrow from a donor that has a marker on the chromosomes that's different from the host? Then I could see if it was donor or host that regenerated the system. And it turned out to be donor. Now that was really important. That happened in 1956. 1961, a group in Toronto wanted to be able to quantitate the radiation damage to normal cells compared to cancer cells, because there was this burgeoning field of radiation therapy of cancer. And they transplanted normal bone marrow into a mouse to figure out how much bone marrow was required to save the mice. Then they could irradiate the bone marrow with one or another dose and figure out how many cells they killed. But when they were transplanting the bone marrow, they found that, here we go, this cell and this cell and this cell and this cell and this cell was made by a single cell in the bone marrow. And the way they could do it is they irradiated the mice. They saw bumps on the mouse's spleen, even though they died because they didn't have enough cells to regenerate platelets for the blood and granulocytes to fight off infection. So they guessed that there must be a cell, a single cell, that could give rise at least to what we call the myeloerythroid cells. And they changed how we thought about it because they quantitated. They found out the more bone marrow cells they put in, the more bumps there were. And then they showed by the cleverest experiment of all that each bump was derived from a single cell. Now it turns out it wasn't the stem cell that we're working with now, but they thought it through well enough to say, well, if you put in cells and they cause a bump, and if you know all the cells are derived from a single cell in the bump, what if you just retransplant the whole bump? So they did. And they found that not always, but sometimes, they could give rise to many bumps from a single bump-forming cell. And that was the beginning of the field of stem cell biology. They proposed, and later we showed, that there does exist a rare cell in the bone marrow of a mouse that has this peculiar property that when it makes 2 daughter cells, on average, one of them is just like the parent cell. Now that is the single and only definition of a stem cell. A cell that, when it divides by self-renewal, makes at least one more cell just like it, as well as another cell, I'll show you, that is one step downstream in its differentiation toward making all the blood cells. So we set up various assays to identify the cell that could make those bumps, what we call the myeloerythroid, another kind of bump in the thymus for T cells, and another bump in a dish that could give rise to what's called B cells. So we were looking for a single cell that could self-renew and make all of the cells in the bumps. We had to have a method of getting the cell. So we made what are called antibodies of a special type called monoclonal. So we could get an antibody from a single antibody-forming cell, and the antibody we would use to bind to all of the bone marrow, which had the blood-forming stem cells somewhere hiding in it. And if the antibody bound to it, we could sort the cells with a machine developed here at Stanford by Len and Lee Herzenberg in the 1970s to sort out either this cell or that cell. Now, I'm not going to go through all of the tests that we did except to say we could eventually purify cells that were very enriched in making bumps, that they could make bumps for the T cell type, bumps for the B cell type, and bumps of that splenic myeloerythroid type. And our objective was to take a bone marrow from a mouse first and then a human that had this thing called HSC, which is Latin for blood-forming stem cell, as a rare cell, but also had cancer contamination, as occurs in many people who have cancers that have spread beyond the original organ. And also we wanted to get rid of these cells called T cells in case we wanted to transplant my bone marrow into Bev. If I had any T cells left in the transplant, they would recognize every tissue in her body as foreign and try to reject her skin just as they would reject a skin graft from her to me. And they would reject her liver just like a liver graft from her to me, or me to her, whichever way it goes. And those T cells that would react against every tissue in her body cause a disease called graft versus host disease. And for the most part, we didn't want to have those T cells there. So we figured if we had one general method to purify blood-forming stem cells, we'd get rid of all those unwanted cells, and we could see if we could do something important. Well, back in 1988, we had purified those cells 2,000-fold. And so our first real test that had clinical relevance was to say, if we transplant these cells, they should be enriched in the ability to save lethally irradiated mice by giving rise to donor cells that make all the blood cell types. That was the experiment we did, and this is the actual experiment from that time. It took 200,000 bone marrow cells, not fractionated, to just save 90% of the mice. It took 40,000 to save half the mice. We were at that time at 1 in 2,000 cells. So if you divide 2,000 into 200,000, you get 100. And that's exactly what we got, was a 2,000-fold enrichment, so that 100 of our cells at that time were equivalent to 200,000 bone marrow cells. 20 to 30 of our cells were equivalent to 40,000 bone marrow cells. So this is the scientific markup of the stem cell in the mouse, and later, as we found, the stem cell in humans. These blood-forming stem cells, when transplanted into the animal, can make blood, and they make blood for life because they self-renew. And in order to get these stem cells, we had to do a lot of stuff, and you'll notice there's some company names there. We had to form a company because in order to do the transplant series I'm going to show you next, it took about $100 million to get there, to be able to do the phase 1 trial. And nobody at Stanford seemed to have $100 million, at least for me or my lab. So this is the molecule names that are on stem cells, and these are the ones that aren't on stem cells. So they're just as important. So with the Herzenberg cell sorter, we could color the antibody that sees this molecule green, this one red, this one purple, this one yellow, and these orange. So we would tell the cell sorter to give us the cells that were, whatever I said, green, red, blue, and yellow, but not orange. When we did that, we could then ask, did we get rid of cancer cells in the transplant? That was our objective. And here was the test. This is the fold reduction of breast cancer cells in what would have been transplanted into a woman, or non-Hodgkin's lymphoma, or multiple myeloma. And in all three of them, only if we use the collection of antibodies that we had defined, not a single antibody, but the collection, did we get 200 or 220,000-fold or 150,000-fold depletion. So the company that I started did a clinical trial in stage 4 breast cancer, in the hardest group to treat, non-Hodgkin's lymphoma, and in multiple myeloma. This breast cancer was done at Stanford, run by Rob Negrin, who was the head of our bone marrow transplant program. So here's scientific language for saying, would the stem cells save women who had stage 4 breast cancer? Now, probably most of you don't know. Stage 4 means that the breast cancer is gone beyond the breast, and usually gone beyond the lymph nodes that drain the breast, and now hit the bloodstream and land somewhere and grow what's called a metastatic or spread focus. If it lands in the liver, lands in the bone, lands in the lung, usually the docs could know that it had happened. Also, if it goes to the brain. So the only exclusion of patients at this Stanford trial were those that had it in the brain, because back in 1996 to 98, the drugs that were available weren't good at crossing the blood-brain barrier and treating the cancer. But that was the only exclusion. They were taken, the first 22 patients in a row. Now, when you see a blue dot here, this is how long it took that patient getting her own stem cells that are cancer-free to regenerate the infection-fighting cells. And the yellow dot is how long it took her to get platelets sufficient number to feel that she was safe. And what you can see is over a large dose range of stem cells, if this thing works, 100-fold dose range, only with the lowest doses was there a prolonged time to get enough platelets to be safe. Any dose from here on out, this was a patient that had an infection and had a little delay, we got stem cells to take. And this is the first time ever that patients got a cancer-free transplant. We checked every transplant, or certainly Rob Negrin did. So that's what happened in that trial. This was called a phase 1/2 trial. Didn't expect to get data other than safety data. But of course, they kept looking at what happened. And so this is up to about 5 years after the transplant in Rob Negrin's group. So this little X here was what was happening all around the world if you gave back what's called mobilized blood, a population that includes stem cells, but wasn't purified to get rid of the cancer cells. And in their study and in our study, about 40% of the women with that stage of disease already had cancer cells in the bone marrow, and lots of them. So 6% alive without disease at 2 years, if they got back mobilized cells alone. And here, 45%. Sounds impressive, but it was only 22 patients. And then overall survival is that way, and at 4 to 5 years, it had dropped to 37%. Now I'm told, but I can't say for sure because it's secondhand, that they're down to 16% at 8 to 10 years. I'll just say this is probably different than this. And the general idea of isolating a patient's own stem cells free of the cancer cells so that you can give an increased dose of chemotherapy or radiation that would kill the patient unless you gave back the stem cells looks to be pretty important. And it also says if only we had lung regenerating stem cells and liver regenerating stem cells, we could focus on therapies that were lethal for all the cells in the liver or all the cells in the lung. So part of the Institute of Stem Cell Biology and Regenerative Medicine at Stanford is to find each of these stem cells and to find out, once you've got them, if they regenerate the tissue. And in the cancer setting, if it allows you now to focus the therapy where you might need to. Those people in the audience who have had colorectal cancer, like I did 20 years ago, are especially worried about the liver because the blood supply from the colon or the rectum goes right to the liver. So if we had liver stem cells, we could probably increase the fraction of people who are doing well, and so on. Now, along the way, we began to understand a lot about the blood-forming system. In my lab, we had isolated not only the blood-forming stem cell, all of the other cells in blood formation. So here at the top was that stem cell. Here at the bottom are the cells of the blood. And every step on the way is a cell type that we've isolated either in both mice and humans. So we know what those cells are. And here's the one big surprise. Of all of those cells, the only cell that perpetually self-renews is the blood-forming stem cell. So somewhere here on the way to these what we call myeloid cells, granulocytes and macrophages, are the cells that could become leukemic and give you chronic or acute myelogenous leukemia. And we didn't know what cell it was, but we knew in normal people only the blood-forming stem cell self-renews for life and therefore gives you regeneration of your own cells for life. Well, we assumed that it was the same for solid organs, that there would be a normal breast stem cell that would give rise to the various cells in the breast. There would be a brain-forming stem cell could give rise normally to those steps. And so if we were ever going to figure out where the cancer starts, which cell is the cell that passes the cancer on from one part of the body to the next, we needed to know as much as we could about the cells in the system. This whole field got a jump start when Michael Clark, then at Michigan, now at Stanford, with Sean Morrison, formerly my graduate student at Stanford, took women's primary breast cancer from their breast and made cell suspensions, and they found they could transplant the breast cancer from a person into the breast or mammary gland of a mouse that was genetically unable to make an immune response at all. So the cells could transplant, but only the breast cancer stem cells, if they exist, could transplant the whole tumor. And here you're seeing one of the markers that they used. So they had an antibody that would see a molecule on the surface of many but not all the cells in a breast cancer called CD24. And if they put 20,000 of the CD24 positive cells into the breast of a deficient mouse, no tumor. But if they put only 200 of the CD24 negative from the same tumor, the breast cancer grew. And it regrew both the 24 minus and the 24 plus cells. And then if they did a second transplant, only the 24 minus. Then they used other markers, one of them called CD44, and that was a positive marker, so that they could actually get down to a small number of cells that would reinduce the breast cancer. So, now we go back to that question. In either leukemias, for the cancers of the blood-forming system, or breast cancer, can we know not only what cell induces or transfers the cancer, but what are the properties of those self-renewing cancer stem cells? So cancers don't come from Mars, they come from our tissues. Somewhere in that developmental pathway, a cell goes very, very wrong to become the cancer. At the same time that Mike was doing solid tumors and published in 2003, we were doing the leukemias. And we knew that we could transfer acute myelogenous leukemia into immunodeficient mice. And we started asking, well, what cell does it? A friend of ours, a colleague, John Dick in Toronto, had said, well, it's in a population of cells that has a couple of the markers that are on stem cells, so he assumed they were the same population as the normal stem cell. But when we isolated bone marrow from real patients, they actually came from Hiroshima Hospital, that had acute myelogenous leukemia, and they had a particular chromosomal abnormality we could follow, we found that the leukemia stem cells were not in the normal stem cell pool, but in the next step down, the cell that normally doesn't self-renew. That transferred the leukemia, and we called that the leukemia stem cell. But more importantly for that kind of a study, we found the chromosome abnormality existed not only in the leukemia stem cells, but in the population of normal stem cell. But here was the difference. The cells that had that first abnormality, a chromosomal translocation, were normal. And in those patients, for as long as they were followed, 150 months, if the marker was in a cell that was the normal stem cell population, it was normal. Yet that cell, which gives rise to that cell, gave rise to a set of cells that had more than that marker change. They had added more genetic changes to get the leukemia. So if we now look across the top, instead of top to bottom, there's the stem cell, there's the cell type it gives rise to, and that gives rise eventually to blood cells. I told you before, the only self-renewing cell in that whole series was this stem cell. Yet, I just told you that the leukemia was at this cell stage, the acute myelogenous leukemia that killed the patient. So that led us to propose that cancer, as many people know, is not only a multi-step process, where one at a time you have genetic changes or heritable changes in what we call the clone of cells. It must be occurring in the normal stem cell pool. So sure enough, in all the leukemias that we've gone back to look at, the first event has occurred at the level of the stem cell, and they compete with the normal stem cells. Now, if at the very beginning we could separate the normal stem cells from the leukemia stem cells, we could treat the patient and give back only the normal cells. And Bev and I are working on that in a pre-acute myelogenous leukemia disease called myelodysplastic syndrome. We're going to try to be able to separate those cell types. Now, once we go through all the steps of adding the second step or adding the third step, not we, but the cancer, the leukemia does, it gets closer and closer to the final step where you have a leukemia stem cell. In acute myelogenous leukemia, I told you it's at this step. I'm going to show you that in chronic myelogenous leukemia, it's at a step downstream. You're going to have to trust me for this. We'll just say that Katrina Jameson and I looked at the last stage of myelogenous leukemia when it's about to kill the patient. And it's in a cell type we call the granulocyte monocyte progenitor. We could transfer that into mice and retransfer that, and it transferred the disease. The blood-forming stem cell that had the early stage, had the genetic malformation, never transferred the disease. And here was the shock to all the hematologists who taught me a long time ago. The cell that tells them it's the leukemia, which they call the blast cell, the big cell, didn't transfer the disease at all because it was coming from the progenitor of the blast cell, which was several steps removed from the stem cell that had the beginning stage. And this is just ways to show it. So we had shown in breast cancer and in leukemia that there was a subset of cells within the cancer that we call, in general, tumor stem cells, that they continue to try to make the other cells of the tissue, and that if you don't get rid of them, they regrow the cancer. But if you could find something specific for that tumor stem cell, we might be able to get rid of the cancer. So that's the whole idea of what we're following. And now we're trying to find ways to test the hypothesis with authentic human primary cancers. Here's one way that my fellows Ravi Majeti and Chris Park did. They took the human leukemia stem cell and compared the genes it expresses with a technique developed here by Pat Brown and over in the commercial community by a company called Affymetrix. And we compared the genes that were on, that's highest level in red, if this thing would ever work, in the leukemia, but not on in normal blood-forming stem cells or normal multipotent progenitors. And to our surprise, there was about 4,000 different genes expressed. So in all that long step to make a leukemia from a pre-leukemia, many, many changes had occurred. And in this case, we could look at those that were on the surface against which we could make antibodies and see if they caused the leukemia. The first one was a gene called CD96, and if we took authentic patient cells, Nelke Hosen did this, and separated them into the 96 positive or the 96 negative, and transplanted them into those immunodeficient mice, only the 96 positive in that patient, not the 96 negative, would transfer the leukemia. Proof that we had the tumor-initiating cell. And it was true not only in that patient, but in the next 10 patients as well. So here's one marker that distinguishes the leukemia stem cell from the normal. Turns out to be a gene that's normally expressed in a population of cells that's supposed to protect us, called natural killer cells. Somehow the leukemia got the expression of that gene, and it was part, but not all of the process. The next one, found first in mouse leukemia by David Traver and Sid Jaiswal in the lab, is called another funny name. CD is a terrible name. Just remember that CD will be in front of a number that specifies a gene. This is a gene that is expressed on the surface of normal cells to prevent another cell that we have in the body called a macrophage from eating them. Young cells, like young red blood cells, have a lot of it, and when it fades away, the macrophage finds out by another molecule that binds these cells. If it had CD47, it can't eat it, but if it doesn't have CD47, it eats it. So we went to human leukemias to see if this was true. Here's a leukemia with CD47, incubated with macrophages, the eating cells. They're the big things here. And you can see if they have CD47, they're not eaten and they grow in a test tube. But if they don't have CD47, every macrophage is chock full of those authentic human leukemia cells, and it eats and kills them. If you transfer the one that has CD47, it spreads throughout the body and will kill the mouse. But if it doesn't have CD47 on it, it doesn't even take. If we took authentic primary leukemia cells from patients and put it together with their own macrophages, the eating cells, and then if we block the CD47 don't eat me signal with an antibody that blocks its recognition, it gets eaten. And this is just a mathematical way of showing how many get eaten. So Mark Chow, a medical student who took a couple of years off medical school to pursue this, with Ravi Majeti and Ash Elizadeh and I, began working on it. Now, Ash Elizadeh is a spectacular oncology resident who got his PhD at Stanford with Pat Brown by working out the gene profiling of human B-cell lymphomas and leukemias. And here he asked the question, well, if I go to look at the database of patients who have the CD47 molecule on at high levels, versus patients in the same study that had it on at low levels, what was their outcome? If you just asked the data, what was their outcome? Well, the 30% of the patients that had the highest level of this "don't eat me" signal on were all dead, no matter what treatment, by 5 years. And those with the 30% lowest, 40% were alive by 7 years. So this is independent of any experiments we do, independent of any analysis we do. Ash Elizadeh showed this is a bad risk factor if you have acute myelogenous leukemia. And so Mark Chow and Ravi Majeti pre-coded authentic leukemia cells with a blocking antibody and transplanted it into those immunodeficient mice. If it was a blocking antibody, the leukemia didn't grow. If it was an antibody that didn't block CD47, the leukemia grew and grew. So then we got to the final test. Transplant the leukemia into those mice without any antibodies. Let the leukemia grow up so it looked just like a leukemia in the patients, and then give them an antibody that isn't against CD47 or an antibody that blocks CD47. And this is the clearance of the leukemia from the blood. And this is the leukemia in the bone marrow of the mice. And this is the clearance of the leukemia from the bone marrow. So this isn't perfect, but this is about as good as it gets, that we have two different markers, two different antibodies, both of which see the leukemia stem cell, but not the normal stem cell. So with Bev, we are going to ask the state of California to help us fund this. And now we have names for each of the events that are going on to move toward the leukemia. So then the big surprise came from Keith Chan, who's in the lab and works with the fellows that Linda Shortlift, the head of urology, got to work with us. And this is a characteristic of what we're doing when we marry cancer stem cell or stem cell biology with real human cancers. These are bladder cancers, and Jim Brooks, Joe Presti, and Linda Shortlift and their fellows got out the breast cancers. Keith Chan found a marker on the bladder cancer that would identify the cell that could transplant into the immunodeficient mouse. And here's the cancer growing in the skin of an immunodeficient mouse, a bladder cancer. There's the marker we use to isolate it, CD44, and it's only on the cells on the outside of the tumor closest to the oxygen supply that are the cancer stem cell, and the daughter cells are growing in. And when we co-stain it with CD47 just on a section, those same cells had the highest level of CD47. So Mark Chow and Keith Chan said, could we make the bladder cancer also be eaten by the patient's macrophages and kill it? And the answer is yes. The antibody to CD47 allows them to be taken up by the macrophages and killed. Now, we didn't kill every cancer stem cell, but we know what was true for leukemia is now true for these set of bladder cancers, and is true for brain cancers, and is true for some head and neck skin cancers, and is true, it looks like, at least the beginning analysis, for ovarian cancer and a childhood brain cancer called medulloblastoma. So what have we done? We've started to look at normal blood-forming stem cells. We've found that we can get cancer-free stem cells, and they will regenerate the blood-forming system of a cancer patient. The second thing is we looked at the stages of development, and we compared it to leukemia progression, and we've been able to isolate in acute myelogenous leukemia and in the final stages of chronic myelogenous leukemia, the leukemia stem cell. Mike Clark and others have found the colon cancer stem cell here at Stanford. Mike Clark and others found the breast cancer stem cell. Nobuku Uchida and Tsukamoto and I found the normal brain stem cell. And Peter Dirks, Jay Rich, and our group Tall, Rivet, and others, have found the glioblastoma and the medulloblastoma stem cells. So we're on the road. And by looking at the cancer stem cells, which we can isolate thanks to work that's been done by the Herzenbergs, looking at the genes expressed by the normal stem cells versus the cancer stem cells, thanks to work by Pat Brown and Lubert Stryer and others, we can now start to ask questions about which are the cancer stem cells. And I'm proud to say, only at Stanford is this a major effort. And by combining the work of the stem cell scientists and the work of people who want to try therapeutically, we hope to move into clinical trials in this and other cancers, I would say, over the next 5 to 8 years, and I hope it's successful. So that's where we are today with cancer and normal stem cells. Thank you. So now I'm going to answer questions. Myeloid really means cells that are growing in the bone. And the myeloid cells we're looking at are the subset of cells in that hierarchy that make granulocytes and monocytes normally, but get diverted to make a myelogenous leukemia. Sorry I didn't say that before. The first treatment we're going to try is we know that the antibody to CD47 makes the cells susceptible to macrophage eating. Now, usually when you try one antibody, there will be in those billion cells that are your leukemia, a cell already that had a mutation, so the antibody doesn't work in the way you think it's going to be. Joshua and Esther Lederberg, before they got here at Stanford, had already shown that mutations pre-exist in a large population. And if you want to kill a bacteria with penicillin alone, it's almost impossible, but if you put penicillin and streptomycin together, the cell that's resistant almost never has dual resistance to both. So we're going to try to make antibodies against CD96 and CD47 and start with that trial. Max Jan and Ravi Majeti are working out a third one. And we found, because we could do those expression profiles, 6 targets. So before we treat patients, we're going to see what synergy exists from 2 antibodies or 3 antibodies at a time, so we can find the right combination that no leukemia has already subverted. So far, it's true. So far. I can't say every single cancer because we haven't looked at enough. But so far, this is a trick that's been acquired by the cancer and the leukemia cells to avoid that first line of defense. Now, after the first line of defense, the molecule actually CD47 says don't eat me to, is a molecule called SIRP alpha, S-I-R-P alpha, right? So I showed that the macrophage, the eating cell, has SIRP alpha. If CD47 is on the cell it wants to eat, it doesn't eat it. There's another cell type that has SIRP alpha on its surface, and it's called a dendritic cell. The dendritic cell is the cell that eats the cancer cell, digests the proteins of the cancer cell, and presents them to the T cells of the immune system. So we suspect that those cancers that have put CD47 on the surface are blind to the macrophages and blind to the immune system T cells. Now, there's a lot of us at Stanford that are working on dendritic cells or T cells. And so we expect to build a team over the next few years that will look at this second part of it, the T cell, and see whether or not we can now render the cancers to give up the information of their new proteins, not only to be eaten by macrophages, but to stimulate immune responses by T cells. And so that's part of what Bev and I are trying to do in the Cancer Center, is to bring in the best T cell oncologists we can find and to work with us on it. But already Ed Engelman and a group at Stanford are figuring out the biology of the dendritic cells. And we actually have the world's leading T cell immunologist, Mark Davis, who's going to collaborate with them to help develop new therapies along the T cell line and we along the antibody line. And with a guy you may have heard of, Ron Levy, we're looking to see if the antibodies will work for B-cell lymphomas as well. Trying to find out some of the secrets of the brain cancer. So the neurosurgery group hired Albert Wong out of Philadelphia to come use antibodies against a particular molecule that most glioblastomas, the last stage of glioma that can kill, and he's working on an EGF receptor, and he has a particular antibody that looks pretty good, and he's working out antibodies to activate that and also to find the cancer stem cell. So he's making antibodies that have two combining sites. Others are looking at those cancer stem cells and saying, just a second, there's a pathway that the cancer stem cell is using to grow. Why don't we see if there are small molecules that block that pathway and see if they block cancer growth? So again, at Stanford, we're following pathways like, these are strange names, the sonic hedgehog pathway, or the wingless pathway, or the notch pathway. These are all what fly geneticists taught us to look for. A fly that looks like a hedgehog, or a fly that doesn't have wings, or a fly that has notch in its wings. And these are genetic pathways of signal transduction. And already, Phil Beachy has found the lead compound to block the hedgehog pathway. And Roel Nusse is working with people to block the wingless pathway, and so on. So small molecule therapeutics are just coming in as well. The answer is so far, not so far as we can tell. It just blocks its interaction with the SIRP-alpha on macrophage or the SIRP-alpha on dendritic cells, so far. Now, some antibodies to CD47 send a signal to the cell to commit suicide, but that happens hours and hours after it's been eaten and killed already. Well, with CD47, it's a real worry because it's used by many brain cells for things we don't know yet. But in the blood-forming system, at least, it's not on the resting blood-forming stem cells, but only gets turned on when they have to go into the bloodstream and regenerate another site that lost its blood-forming stem cell. So that trip that it takes, which I removed the slide, the blood-forming stem cell gets out of the bone marrow, gets into the blood. It uses several genes to get there. It's in the blood for about 1 to 5 minutes until it sees a blood vessel that has the target that it homes to. It uses integrin alpha-4 beta-1 to find a molecule called VCAM1 on the surface of the blood vessel that's only in the bone marrow, liver and spleen, which in the mouse is the blood-forming organs. In the place that has the VCAM1 positive blood vessels, it's lined by macrophage. So it's got to get past there with CD47, and then it has to get back in. Well, the normal stem cell only turns up CD47 when it's in the travel mode. So anti-CD47 antibodies deplete only those stem cells that are traveling at the time you treat it. But we still have to go look at every tissue to see if it's toxic. Now, we have done the trial in mice. with the antibody to mouse CD47, and we know which cells to look at, and amazingly, so far, the mice have not keeled over with the antibody to CD47. And the antibody doesn't get in the brain yet, so it doesn't cause the mouse to get dumb or stupid or whatever it is for the brain function. So far, so good. So when we talk about taking a lead like this to a therapy, it means that we do 3 or 4 years of preclinical testing to see if we're going to get away with it, not to cause damage to the normal cells. I wish I could say that they do. But in fact, when we look carefully, the drugs that have been developed so far that shrink the tumor down by 90% don't shrink 90% of the cancer stem cells so far. Already we know that most of the stem cells, whether they're cancer stem cells or normal stem cells, they have a pump in them that pumps out drugs very efficiently. And their daughter cells here that are killed by a drug don't have the same pump. Just recently, Max Dean, working mainly with Mike Clark and a little bit with me, has looked at cancer and normal stem cell and says, well, what about radiation? We use radiation to try to kill cancer, and it kills the daughters of the cancer cells much more efficiently than the cancer stem cells. It kills cancer stem cells, but they got a protection against radiation or against DNA damaging agents. They make a molecule or a set of molecules that sop up the oxygen-free radicals that radiation induces that causes DNA damage. So Max Dean, a radiation oncologist, says, I'm going to try to find drugs that take away from the stem cells their protection. I'm going to give the drug whole body, but I'm only going to radiate the tumor. So I'm going to take away the way the tumor stem cell protects itself against radiation, and see if I can reach a therapeutic dose. So Max Dean is right in the middle of those trials, and we're right in the middle of trying to keep Max Dean at Stanford. Thank you.
For more, please visit us at stanford.edu.